So I'm just going to give you a very quick 5-minute update on the latest solution with Oxford Nanopore, and then we're going to go to the exciting talk from Ray today. I think we probably all agree that in the last 5 to 8 years, single cell and spatial methods have really revolutionized the field of transcriptomics. And I think what's driven that is the new biological discoveries that have been made. But on the technology side, this has been enabled by new technologies that have made it much simpler to analyze many, many cells, tens of thousands and hundreds of thousands of cells. So what we've seen is that the scale of single cell experiments continues to grow. And part of the reason for that is simple, that the more cells you have generally correlates to more resolution and more biological discovery. So there's one axis of resolution, which is increasing the number of cells per sample. But what I want to highlight briefly is that there's another axis of resolution, which is not very well addressed by just adding more cells per sample. And that is when we go to sequence our single cell samples, how much of the transcript do we actually read? So here you have a figure from a publication, and on the top we have an annotation of a single gene. In the middle panel, you have a typical short read data set with the reads piled up at the 3' or the 5' end. And this data set, this type of data set is really useful for doing gene expression, extracting gene expression information with lots of reads. But if there's something else further down in the transcript that's important to the biology of the questions that you have in mind, that's probably missing or difficult to extract from this type of data. On the bottom panel, you see Oxford Nanopore reads, and you can see that each of the reads has an isoform contained within that single read, and you have the direct exon-exon connectivity present there. So there's potentially a lot of things that are missing when we only sequence a small piece of each transcript, but I just want to highlight the top 3 reasons that we see people switching over to long read sequencing from single cell and spatial assays. The first we already mentioned on the previous slide, which is alternative splicing. And I want to highlight that in particular, neuro and brain cell types where there's a lot of splicing, this is where we see a lot of use of long read sequencing. Isoform switching is another one where you have the same gene expressed in 2 different cell types, but the isoform is different in those 2 cell types. And we see a lot of use of this in developmental biology. In the middle column there around discovery, I want to highlight the main application in cancer, which is often actually around doing genotyping. So looking at SNPs in the genome that are carried through to express transcripts. And often in cancer, the ability to pull these SNPs out of the full-length read, so SNPs anywhere in the expressed transcript, is as important as isoforms, or even more important. The ability to look at things like allele-specific expressions and fusions as well. So those are the top 3 reasons we see people using Nanopore with single cell methods. And so with the latest chemistry and solutions from Oxford Nanopore, you can do now isoform level clustering. And on the left, you see a very familiar plot. But when you do this clustering at the isoform level, in some cases, you see differences that are hidden at the gene level. And on the right, you see a 10x Genomics Visium spatial assay. Now you can break this down into isoform expression. Look at specifically here in the mouse brain, certain transcripts of this SNAP25 gene that are expressed in different regions. Lastly, let me just give you a quick overview of the solution for sequencing on Nanopore from 10x Genomics cDNA. First of all, I want to highlight that the prep on the left there uses the standard 10x Genomics output, so you don't need to change anything in your 10x assay. You take the full-length cDNA, and you don't need to split this sample into multiple tubes. You use one set of tubes per 10x genomic sample, and it actually only takes about 3 hours to go from 10x cDNA to be ready to have a library that's ready for sequencing. Now, the nanopore sequencing solution, many of you are familiar with, but it's a very different technology to others out in the field as well. So we don't use sequencing by synthesis. We don't do cluster amplification. And because each nanopore sensor can read thousands and thousands of molecules one after another, we don't do any kind of concatenation, and we can still utilize the system efficiently. The Promethean platform also has a range of devices that allow you to sequence 2 samples at a time, up to 48 10x genomic samples in parallel. And you don't need to wait to batch those samples together in most cases. On the right-hand side for data analysis, because Cell Ranger is not quite yet set up to give you isoform output, we provide a free tool to do the demultiplexing of your nanopore data. And you don't need to run any short read data in parallel. This runs on nanopore data alone. Lastly, for a little bit more detail, the kits that are certified compatible with 10x Genomics are their 3', 5', and Visium spatial kits. We're also testing their new GEM-X chemistry in-house, which is looking really good. You use 10 nanograms of 10x Genomics cDNA to do the library prep. And in the middle column, we recommend sequencing every 4,000 to 5,000 cells on one Promethean flow cell, roughly, to match 20,000 reads per cell guidance from 10x Genomics. And we expect an output of around 80 million cells for each flow cell. And the output that you get from our tool will give you cell by gene that you're used to getting from Cell Ranger, for example. But you'll also get cell by isoform count matrices, as well as a tag bound file and a couple other useful metrics. So hopefully that was a useful update on the latest from Nanopore. And now we're going to go to our scientific talk from Ray, and I'll pass over to him. Thank you.
Thanks, Daniel, for the introduction and a wonderful talk on the background of the Oxford Nanopore. So my name is Ray, and today we'll just share with you some of our experience using long-read sequencing for a single cell. So our lab is interested in understand the human eye, particularly the back part of the eye called neuroretina. This is the part of tissue where we receive the photon, the light, and sense it and convert to electric signal that we convert that we transmit to the brain so that we can see things. So the reason we like we're particularly interested in this particular part of the eye is this is one of the tissue that is the main target for erasable blindness. And then if you have degeneration in this part, and there's now there's only very limited treatment to reverse it. So we would like to learn more biology of this so we can be better develop therapy in the future. So the retina even is only compared to brain is much simpler. It has 3 layers of neuron cells, and then however, despite the simplicity in the anatomy, the number type of cells in the tissue is still enormous. So the single cell RNA-seq profiling has been done on mouse retina, and then this is the latest map representing the atlas, and this is composed of about 300,000 cells RNA-seq, and then based on this data, it can be divided into 120 cell clusters. Each one, most of them will be corresponding to distinct cell types. So they have different color as different class, and where each cluster is different cell type. So this is enormous data, and that'll be really useful for the community. But one, as Daniel mentioned before, there's major information missing, and the limitation is because this is from the 3' RNA-seq, primarily from the 10x genomics data. And then therefore, we really don't know the isoform. And then we know isoform is important during development of the retina. There's significant isoform switch during development, but also we also know as neuron, they actually use a different class of isoforms and then to basically build up the map. So isoform has been known to play an important role in development and function of this tissue. So to identify those isoforms, we design a pilot project before we want to scale up, in particular in the human tissue. So we would like to basically perform some long range sequencing, use Oxford Nanopore, and to evaluate its performance against the the transcriptome profile at the single cell level against the short rates. And then we also would like to, during the course, to identify mRNA isoforms and generate a catalog in the retina at the single cell level. And finally, we would like to understand the distribution of this isoform across different cell classes, subclasses, and types. So here's the study design for this pilot experiment. So we start with the mouse samples, and then the retina we dissected, and then we perform, the regular standard text genomic single cell isolation and then profiling. And once we get the library, then we'll actually split them into two half. One is go to the normal Illumina short rate sequencing, and then we'll perform the UMAP clustering and annotation. While the other half we convert to a library that is suitable to be sequenced by the Oxford Nanopore, and then we perform long rate sequencing. So once we perform these two sequencing, the short rates short rate sequencing data will be used to cluster and then identify cells barcode that go into this sort of captured, and this will be used to guide looking at long rate data and then for cell barcode and cell identity assignment. So once it's that done, then we will basically aggregate this short mini bulk, right? For all the long rates belong to the same cell type, then we aggregate together and all the rates, and then we perform basically alignment and then isoform identification and categorize the long rates or the transcript into different category. And then we can compare to some downstream analysis looking at the distribution and what type we get. So we'll walk you through these steps later on. So this is the data. So for the short rates, so we pick up 4 different retina. The reason we want to pick these 4 samples is there are 2 or sort of replicate. So this one and that one is basically wild type retina, and then this particular CD73 depletion, and then the amacrine cells are the 2 samples that are specifically enriched particular cell type, rare cell type in the retina. One is for the bipolar cells, like this one, and then one for the amacrine cells. So you can see the distribution of the cell are quite different. So that way we can cover both the abundant cell types, like here in the rods and the cones, and also the rare cell types. So in total, in this short rates, we generate about after filter, we have 1.5 billion rates total. And then use the same and the second half of the library, and then we perform the long rate sequencing, the same 4 tissues, same 4 libraries, and then we generated 1.28 billion raw rates, and after filter and then assignment, we got the half billion left. So looking at the cell clustering, so we look at the single cell sequencing from short rates, and those are, as you can see, this different color corresponding to different different samples. As I mentioned, the 2 samples that are from wild type, they actually all sort of aggregate together here, like this part. These 2 are really go together because that's what we expected. And then the other 2 samples, which are representing different cell type, actually separate differently. And this is just looking for cell types and clusters. So you can see that's what was expected. So and then similarly, looking for long read data set, you also see the green clusters are coming from the bipolar cells. The red ones are amacrine cells. The 2 wild type go together nicely, and then those are different cell types. So again, this also they perform as we expected. So then we start looking at the concordance between the short read and long read data set. And then those are annotated at different cell class. And then you can see those are the marker genes. And then the bottom line is the marker gene we know for cell type, cell class specific, it show very similar pattern. So they're pretty consistent. So we can look at it more closer. So this is the high concordance we are observed. So this is short rates and long rates, and then you can see the percentage expression of the key markers are very similar. And then this is the concordance of assignment of the cell are very similar. So if you look at just this sort of confusion matrix, you can see especially 90, most of them are 98, 99% identical, and except this sort of RDC cells have slightly dim results, and then because they are much rare cell type in our data, so this is more fluctuant. So then we can now integrate the co-embedding the short reads and long reads together, and here's what you see. And you can see they go very nicely together. Red is the long reads, the green short reads based on top of each other, and then those are cell types they identified. So very nice cluster identified. And then if we zoom in even further, this is the cell type shown here, and then you can see this is the bipolar cells, and this is the rod cells, and those are the Amican cells, right? So as I showed before. So then we look at the marker side by side, and you can see the pattern for different major cell class are essentially identical. So this is the long reads and short reads, the long reads and short reads, like that. You can see they actually align. Pattern are very, very similar. And then we can look closer for the feature plot on the integrated map, and then you can see this is long reads and short reads data, and this is the different markers for different cell type, and then you can see the pattern are essentially identical. You can also look at the von Weyl plot. You can clearly see, for the markers, the orange is the long reads, and then the green is short reads, pretty much the same. So therefore, from the long reads and short reads, by looking at cell class identification, cell cluster, and the cell markers, and these two perform basically highly concordant and very similar to each other. Right. So this not only they can look at the cell class level, but then when you zoom in, remember some of that will come from the rare cell type, for example, bipolar cell. Bipolar cell together account for 10%, roughly 10% of the cell population in the retina, and they can be divided into 15 different types. And then when we look at, so when we use high resolution to perform the cluster analysis, we, both data set can identify all 15 types, and then you can see the concordance here are also very high. So it's not only class level, which is very much easier to identify, but also at the cell class level. So now we advance move to the second step, which is looking for isoform. So can the library made from 10x standard flow capture the isoforms? So so you can see, for example, when this is one example, the PCAR KCZ is one of the marker genes for the rod bipolar cells, and have about like 20 exons from the annotation, and then you can clearly see there are two isoforms here, and you can see basically the isoform are captured, even the long one and short one are all captured by different data set, but it seems to have different usage of this isoform. So for example, the Amican cell seems like do not use much of the long form, while the the bipolar cells have some both long and short form, while the wild type, which is pretty photoreceptor dominant, are pretty much have the long form. So from this example, we already can see that first of all, the isoform, the full length can be captured, multiple isoform can be captured, and then the differences of them appears to be also captured. So and then we start to basically, as I mentioned, that for each cell type, then we can aggregate all the long reads together, form this sort of mini bulk sequencing, so to speak, and then we will use that to identify new isoforms. So we perform because this is the first time we did this, and at the time is and then we really worry about potential artifact and this is like the nature. So therefore we use implement very stringent steps to remove all the potential artifacts, for example, internal priming, internal retention, degraded RNA, so on and so forth in our data set. but also require significant abundancy. So we want to basically UMI count, less than 10 are less or equal to 10 are removed, and then the isoform for each isoform need to have at least 5 or more, more than 5 UMI identified. And also we remove, this is actually a big removal if the isoform are less than 2% of the overall abundancy for that gene, then that also be removed. So given the criteria we're using, and then I would say the number isoform identified is probably underestimate, particularly for rare ones. So this is just give you the nomenclature going forward. So based on the annotation and how the isoform or transcript are mapped to the annotated, so they are classified in like four category. One is the full splice match, meaning they match perfectly with the annotated transcript, called FSM. And then we also have the complete splice match, meaning they have the exons and then the junctions as annotated, but they are slightly shorter than the full length. So they are sort of partial, probably like RT didn't go all the way, so it's partial removal. And then so those are two categories we consider are known transcript. And then you have the novel ones, is novel in category, meaning they have the same exons as annotated, but their isolation, the junction are not observed or annotated before. So for example, you're skipping an exon or something like that. And then also there's novel not in category, meaning not only the junction are different, but also just exon are not annotated before. And then the last thing you have fusion, meaning two transcript that span two different genes. So things you can remember quickly is like the green and then the blue are basically known isoforms. Then the yellow and the gray are novel ones, and then the purple, the orange are fusion. So by looking at this category distribution on the isoform we identified, so this is across different cell types. You can see the more cell we have. For example, in this data set we have more rods, and then AC, BC are the one enriched, so we have more cells profiled. The number of total isoform identified is higher than the cells which are less represented. But if you normalize the ratio, and then pretty much 30%, around 30% of isoform are novel, those are yellow. And then you can see the blue and the green account for about 70%. So 30% of the isoform we identify after all this filtering process are considered novel. So compared to the novel isoform and then the non-isoform, here like the novel is blue, and what we find in general or average, the expression level is lower for the novel isoform compared to known, which is expected, right? The known ones have been seen before from previous study tend to be highly expressed. So, and also in terms of the length of the transcript is slightly shorter than the compared to known. And then if we break up to individual more fine, a category of different isoforms, and again the same thing, the NIC, the NNC are based, the N is yellow, and they are in general lowerly expressed compared to the known ones, and then the length of them depends on which category, may not be necessarily shorter, but in general slightly shorter. So how those isoforms are distributed, I mean how many genes we identified, we observe multiple isoforms. So the bottom line is those are the genes that we only observe. So those different cell type, one isoform in the cell type, different color represent different cell class. Well, but you have many have 2, 3, 4, 5, or even more. So if you add this together, so about 70% of the genes we do identify 2 or more isoforms. So most of the genes in our genome do have alternate splicing, and that's actually known, right? Because when we did when that's like even 10, 15 years ago when people do short read sequencing and then cover it. So the conclusion is like 90, maybe 90 or 95% of the gene in the genome have different isoform. But still amazing to me is that this by remove all the slightly low expression isoforms in our filter stage, and then look at just the eye retina, just one tissue, very simple tissue, and then we already have 70% of gene have different splicing. So the isoform is actually very abundant. So the next question we want to ask is, okay, now we know most of gene, 70% of gene have multiple isoform. So do they really show cell class specificity? I mean, how are they distributed? Do you see one cell type express isoform 1, the other cell type express isoform 2? So then we look at through the distribution of this different isoform across different cell types. So the conclusion interesting is that most of the isoforms are observed in all cell class. There are cell class specific ones as I highlight here. And so they do exist in our data set, and however, they are rare compared to the rest of them. So most of the isoform exists in multiple cell types. They don't show cell class specificity. So this is just give you an example to see what it means. So protocatenaria is a gene that have a lot of, it's a gene family, have like 3 subgroup, and then they call alpha, beta, and gamma. And then in the alpha and the beta and gamma, you can see they have a shared exon on the 3' end, but the 5' end, they have a different use of the exons. So like these guys, right, and those guys. So and then they give you, by the combination of alpha, beta, gamma, they give a huge, and beta is quite different. They have single exon, but they just have a bunch of like paralogs. So by have this combination, they can, and these are the surface molecules in neuron, and then you basically have a fingerprint, right? And so they can do repairing of the one or itself exons, but also traction of a different cell exon, because they have expressed this slightly different code. So that's basically the model. So when we start looking at the alpha, which you have different isoforms like shown here, you can see from different cell class, and they pretty much express all those isoforms, right? However, you notice the ratio of it are quite different. And then when we zoom into the bipolar cell itself, like this class, and again you see the similar trend. Different bipolar cell types, although they are very closely related, I mean they express all this isoform pretty much, but the proportion of them are quite different. Similarly, for the gamma, I have the same story where the different isoforms are expressed in all cell class, but between different class, the ratio are quite significantly different. So, and then now, now we basically, the isoform, even we apply very stringent criteria, then we still worry about how can we validate this, right? Because this is really still very large scale, and then you really based on the isoform, based on sequence and alignment. So so we would like to be able to do some sort of cross validation of the long range data. So we then take the retina and perform the bulk RNA sequencing, and to about like 200 million reads, so quite deep, and then from there, and then we can get the perform the and this is the not the single cell. This is just a bulk RNA sequencing. You can cover the region, and then so what we then look at is the discovery. So this is from long reach. We can see again we see this about 30 to about 40% we see are novel. So meaning it's sort of consistent with the single cell, about 30-40% are novel, and that's our distribution. And you can see again, large of them have multiple isoform. So then we take the isoform identified with the bulk sequence and compare with the single cell and see what happens. So what we find is that about 30% of the isoform we identify from single cell can be found in this bulk data set, right? So and that's this is the comparison, and then in there you can notice a lot of them are green, meaning they are known, but they do have some novel also also like basically overlapping, meaning it can be confirmed using an independent sample, independent experiment. So, and then if you look at the isoform that are unique to single cell, and then you can see the proportion of the novel is higher. In general, for the isoform unique in the single cell, and have a lower expression compared to ones that I find in this both bulk and the single cell. Because in the bulk, first of all, the overall coverage is slightly lower than the single cell when together, but also I remember in the single cell, we actually isolate the rare cell types, and then that they will be underrepresented in the bulk data. So I think the combination of these two and then allow you the single cell data actually discover more novel sequencing. But in any case, you have 30% of transcript do overlap between these two data sets. So lastly, I just want to mention that something slightly surprising to me is we during the process we do find about 1,000 fusion transcript identified from this data. So typically when I think about fusion, typically I was thinking about cancer, right? Cancer, there's translocation, there's fusion transcript is critical for oncology and something like that nature. So that's what I learned. But in the normal tissue, I don't expect that happen a lot. So, but nevertheless we do find a 1,000 fusion transcript, and then there's different scenario where some fusion essentially are basically fusion of the genes next to each other. So they pick up exons from this third one, and then they pick up another exon in the second one, and then they jump to the first one, right? So it's like take neighbor exons for somehow the splicing. So meaning initially there's some RNA like this, and then splicing. So the RNA almost like transcribed from here. So I don't know how can I make it, mostly from this 2 and 3. But you have something odd is where you have skipping. You really skip from this gene. You really come from this gene and to the next gene, right? So you skip some genes in the middle. So in general, the transcript of the exon number of the if you compare the adjacent gene and non-adjacent gene, the adjacent gene tend to be longer. Speaking, you really only take a few exons, so they don't make the transcript significantly very long. And then you see the most of the fusion is actually come from the fusion gene next to each other. So are they just like a biological system is not perfect, right? So can you imagine why it happened? So it could be noise. So then we look at the fusion transcript compared to the transcript or genes involved, contained in that transcript. Their normal transcript, so to speak. Just look at the ratio, and then we reason that if it's noise, then it should be a very low ratio. So which is indeed the case. Most of the fusion compared to the ratio to the normal transcript are very low. However, you do have some transcript fusion is actually turned out to be the major transcript isoform, so which is quite interesting. And then we take this one to look at the ontology, and then interestingly, a lot of them are found related to immune system. So lastly, we're wondering like how much sequence do we need for isoform identification with the long range so that we can design our future experiment. So when we look at, we perform basically the simulation by subsampling the rates from 1% all the way to all the rates we have. And you can see in general, for all, isoform type category, known and unknown, the more rates you get more. However, the rate of, saturation is different. So you can see for the known, for example, for the known isoform, which is this guy, the increase from the 50% to all is not that significant anymore. And while for the novel ones, they still keep going up. So basically, if you want to identify pretty much known or more abundant, transcript, then you don't need as much data as we have produced, and you still can get also major isoform distribution across different cell classes and single cell. But if your interest is looking for novel isoforms, particularly the ones in rare cells or low expression, then you still need to, our current data is not saturated. So it's a quick summary, and hopefully we answer the goal we set up to do. We find the long-range single-cell RNA sequencing is actually highly effective to identify isoforms, to identify cell clusters that have very high concordance with your rates, but also identify isoforms. and then compare, this small pilot study we did is actually compared to the reported isoform that people have accumulated over the years. It's an 80% increase from current number isoform. That's actually a significant increase. And as I said, we use pretty stringent filtering, and then the true isoform we have in retina is actually much higher than what we report here. So basically, there's not a lot of isoform to identify. And the second thing, the cell class specific isoform is rare. So, but still, we have 2,000 isoform identified in this data set. This could be used as potentially cell type marker or cell class marker going forward to understand why the splicing is cell type specific. So that'll be really useful tool. And also maybe some of them play a very important biological role specifically for that cell class. That'd be interesting to follow up. So we also identifying the isoform exhibit differential gene expression across cell class. Even most of the isoform expressing across different cell types, but the ratio of those isoform abundance are quite different between cell type to cell type. Finally, we think if you primarily focus on known and abundant isoforms, then the sequence depths do not need to be super high if it's the long rates. But if you're interested in looking for novel ones, then you do need a very deep sequencing. So with that, I just want to acknowledge Meng and Su and Yumen, Jin, who actually conduct the work and analysis and generate the data. Our lab is fortunate to be funded by various foundation and then the institute.